Hi, my name is Odysseus and I'm a paleobiologist at the University of Oxford in the United Kingdom. And today I have been invited to tell you a little bit about Coccolithophore biodiversity, discuss what we use for species concepts and understanding those, and showcase examples of Coccolithophores that were recently discovered and described. But first, a little bit about myself. I come from Greece, alas, not Ithaca, but Athens, and I started out as an undergraduate student in geology at the University of Athens. I moved to France for my master's, and then, faithful to my poleward expansion, I moved into the UK, where I was fortunate to be part of the micropaleontology lab in University College London, and finally fortunate to start my PhD in Oxford, where I have been ever since. I have a strong interest and enthusiasm for diversity, biodiversity in general, but specifically diversity of eukaryotes. And as a community, we have a good understanding in general of the large animals, such as mammals and birds, even if more is needed. But the smaller we go, the more species are left to find. So why is it important that we know and we study eukaryotic diversity? Well, microbes are really essential for maintaining our ecosystems healthy. They provide the food we eat, such as those that enrich the soil where crops are growing, to the pollinators that give fruit, and fish that is used as protein for 1 billion of people. Many of the medicine that is used today also originate from fungi, plants, and microorganisms. And in addition to all this, microbes are the first ones to respond to climate change. And as this continues to add pressure to marine life, there is an increasing fear that species of microbes could be lost before we even discover them. But what do we use to tell that the species is new? What do we draw on for telling differences between the species? And how do we make taxonomy simpler and easier for a more accurate assessment of the diversity? And for these questions, I employ a range of techniques that I will briefly introduce here for Coccolithophores specifically. So Coccolithophores are a fascinating group of eukaryotic unicellular algae. They are haptophytes and they can reproduce asexually or sexually. Well, on the one hand, we have a diploid cell that has the normal DNA complement, and through meiosis gives birth to 4 haploid cells, cells that they have half the normal DNA complement. And 2 haploid cells fuse together and give birth to a diploid cell. The diploid cell is associated typically with heterococcoliths, whereas the haploid cell usually bears holococcoliths, the 2 different types of coccoliths that I will discuss briefly in a minute. And the alternation from a haploid to a diploid phase, or vice versa, is typically captured with a combination coccosphere, meaning a cell that is found bearing coccoliths characteristic of both phases. Coccoliths have been the prime tool for understanding and quantifying biodiversity, both of the individual liths, the coccoliths, but the coccospheres, the whole skeletons as well. And we have 3 main categories: the heterococcoliths that are typically circular to elliptical discs with radial arrays of elements, to the holococcoliths that are produced with mini rhombohedral crystal units, and the nanoliths that lack the features of the 2 and are more uncertain in terms of origin. Molecular genetics is an additional important and powerful tool for telling relationships between the species, and a number of Coccolithophores have already been successfully isolated and cultured and have undergone DNA extraction. And this provides a new avenue for reassessing morphology-based taxonomy and seeing how relationships and species concepts can be assessed or reassessed. And lastly, I would like to mention the fossil record here as well, because this is the only natural laboratory into understanding the ways species and ecosystems experience events and how they eventually bounce back. what do we do for maintaining biodiversity on the planet does rely quite a lot on fossil record as well. And coccolithophores have an abundant and continuous fossil record with the origination in the late Triassic and an increasing diversity pattern up to the KPG boundary, where we have the extinction of approximately 90% of species of them. And then a general increase and decrease throughout the Pleistocene, with a very important element here that the Pleistocene fossil record, the recent fossil record, and the combination of this with the extant representatives, living organisms, can tell us more about evolutionary history and paleobiology of the group. And here I'm going to show a few examples of coccolithophores briefly, covering hopefully a good extent of the diversity today. With the first, the Gepharocapsa huxleyi, one of the most well-known and studied coccolithophores, where through a combination of morphology, genetics, paleontology, fossil record, and ecological data, we came to the realization that this entity represented at least 3 different genetically delineated species, and with a number of subspecies underneath that they vary in terms of calcification, in addition to other parameters. Molecular genetics sometimes is not available for coccolithophores, especially those that have been rare and not easily isolated in culture. And one of them is Calceopappus so far, where the differences in morphology have been sufficient enough to justifiably separate species between themselves. And this is an example of a new species, Calceopappus curvus, where we established it as a separate one on a combination or on the basis of combination of differences across the cocosphere. Moving from heterococcoliths to holococcoliths, even if the haploid phase is more fragile, and this is not typically preserved in the fossil record, holococcoliths and the combination of the types of them on the cell can be a very important feature for telling differences apart. And this, the Anthosphaera type, is a study we did where we understood that the combination of the two types of coccoliths is important for telling differences on the distinct from species to species and other taxonomy units, but at the same time, it suggested that all of them represent a natural grouping on their own. 2 of these forms were found in association, life cycle associations with members of a diploid phase Coccolithophore, and therefore the taxonomy had to be revised and simplified. And the last example that I would like to show very briefly is nanoliths, the ones that they lack the features of heterococcoliths and holococcoliths. And a particular example is the new family Erisiolacea that we established in early this year, 2024, that includes species and genera that have a 3-fold symmetry pattern and a radial growth of elements, which is a feature that is unique in the realm of modern coccolithophores as currently known, and suggested that these forms have an affinity between themselves. So just to sum up, I hope that I have shown even briefly that coccolithophores are very beautiful organisms and very diverse morphologically. We currently have about 300 species, but there are many more to be discovered and described. And integrative taxonomy is the best approach for doing that. Lastly, I would like to emphasize that even if all of the research on coccoliths is exciting, taxonomy is the fundamental discipline and should be taken very carefully into consideration about all other exciting research, as it gives us the background for what we talk. I would like to say a big thanks to all of my collaborators on this, and to the INA Outreach and AURORA Science Communication offices for this kind of invitation. Please do feel free to get in touch if you're interested in this, and thank you very much.